Slides here. My name is Marty Grassi. I'm CEO of Gradient Denervation Technologies. Gradient is a Paris-based medical device company operating within the Sofinova MD Start incubator for medical device innovation. Gradient is developing a catheter-based solution for pulmonary artery denervation. This is a therapy that's emerged over the past few years to treat patients with heart failure and elevated pulmonary vascular resistance or pulmonary hypertension. This is a large unmet need with very high mortality, morbidity in patients, high symptomatic, and patients are in and out of the hospital very frequently. Gradient is a device that is designed specifically for the pulmonary artery anatomy, which comes with its own engineering challenges. We are building on early feasibility data from other devices that have been repurposed from other indications and shown a signal of efficacy, and now we've built a device that is built for this anatomy specifically. We have licensed foundational IP from Stanford University with granted method claims that give us a very nice platform upon which to operate and build. The company was founded within Stanford Biodesign and came to MD Start in 2020, has been incubated since then with funding to prepare for our first-in-human studies. We're raising a Series A financing now. This will fund our European first-in-human and early feasibility studies in the United States. At a very high level, the pulmonary vascular resistance within heart failure is caused and maintained by a link between two parts of the anatomy and physiology. On the one hand, the sympathetic nervous system drives overactivity within the distal pulmonary vasculature. On the other hand, this causes vasoconstriction, which then pushes blood back to the right ventricle, making the heart work harder, leading to the progression of left heart failure into right heart failure with significantly worse outcomes for patients. The data you see on the right-hand side of the slide is an example of this. This is showing patients with heart failure and preserved ejection fraction stratified based upon pulmonary vascular resistance. The top line in both graphs is those patients with heart failure and preserved ejection fraction alone. The bottom line is those patients with heart failure, preserved ejection fraction plus elevated PVR. And you can see a significantly worse mortality and hospitalization. To call out one data point on the slide, there's approximately a 50% mortality rate in these patients at about 4 years after diagnosis, so an incredibly serious disease. We are focused initially on heart failure with preserved ejection fraction. We believe, based on early data, that this would also work in heart failure with reduced ejection fraction. However, there are drugs and devices that work in those patients, in contrast to heart failure with preserved ejection fraction. Therefore, we're focused on HFpEF first. That represents about 50% of heart failure patients. And then from there, somewhere between 30% and 50% of those patients have elevated PVR. There are no approved treatment options. In fact, the drugs that are approved in so-called Group 1 pulmonary hypertension without heart failure are contraindicated in this patient group where heart failure is present. And finally, this represents a huge economic cost for the systems. To put some numbers to this, when we start with heart failure and then progress down to those patients with HFpEF plus elevated PVR, we're talking at about 2.5 million patients between the US and Europe. This will double approximately when we include back in those patients with reduced ejection fraction. At a basic physiology level, what I'm showing here is some work that was done in 1980 with a large animal model for pulmonary hypertension. And what you can see is the researcher was able to create this model and then induce pulmonary hypertension in these animals, and then through both chemical and then surgical denervation, show that he could turn this off, proving the link between the sympathetic nervous system and the pulmonary vasculature. Since then, there's been a significant amount of work in the space in both human and animal trials, again proving that this pathophysiology is able to be affected by a denervation procedure. Two trials that I'll talk specifically about. One is the PADN5 study. This is done by a company in China called Pulnovo. Pulnovo has a radiofrequency device with electrodes around the circle, around a loop type of a catheter. They enrolled 98 patients in Group 2 pulmonary hypertension, or those with heart failure. They randomized 1-to-1 against a sham procedure plus a vasodilator called sildenafil, and they showed a significant improvement in both 6-minute walk testing and a separation in the risk factors that these patients have met within their heart failure groups. The other study is Trophy-1. Trophy-1 is done by an Israeli company called Sonovi. Sonovi uses a catheter that's been repurposed from a renal denervation indication. They enrolled 23 patients with Group 1 pulmonary hypertension at centers in Europe and Israel and showed improvements in both hemodynamics, 6-minute walk, and the number of risk factors that these patients have met. So it's based on this preliminary work that we are now moving forward with our purpose-built device to perform pulmonary artery denervation. The design inputs for this purpose-built device are several, and as I mentioned before, there are some unique challenges from an engineering perspective to build the device for this anatomy. Number one is understanding where the nerves lie. We know that from published literature that the nerves lie around the pulmonary arteries in the main, the left, and the right. These bullseye graphs illustrate the nerves from cadaver studies in patients with pulmonary hypertension. We know that more than half of the nerves are within about 5 millimeters of the vessel wall, and the average depth is about 2 millimeters. From an engineering perspective, there are three main drivers, design inputs for designing our device. Number one, the vessel wall is very thin. It's down to about a 0.5 millimeter thick in many places, kind of a papery thin type structure. This has implications for the energy source that we choose, leading us away from sources that require intimate contact with the vessel wall to transfer heat. Number two is the large diameter range. We know the range in the anatomy that we care about is between 1.5 and 3.5 centimeters. So these are big vessels. And number three is there's significant motion in these vessels. We're right next to the right ventricle, so there's significant cardiac and pulmonary motion requiring stabilization in order to get a consistent thermal dose deposited into the adventitia and connective tissue around the vessel. So we think we've solved these problems. We've frozen the design of our device. We have a size-adaptable device that fits this entire range of diameters. We're using an energy modality that preserves the intima and does not damage it. We are doing our preclinical safety studies, and we're on track for a first-in-human in the beginning of next year. We have a team that has extensive experience developing interventional cardiology and electrophysiology devices, and we have the expertise internal to the company to allow us to move forward into our first-in-human. We anticipate growing the team in the new year. From an IP perspective, I mentioned before we've licensed IP from Stanford University. We have very broad granted method claims on pulmonary artery denervation and feel confident in our ability to move forward. And we're now developing our own company-owned IP to cover our specific device embodiment. A clinical perspective, I mentioned we're pursuing a first-in-human in the Republic of Georgia in the beginning of next year. This will be 10 to 20 subjects with HFpEF plus elevated pulmonary vascular resistance. We're utilizing our physician consultants to help us both with screening and execution of these procedures, and we're working with a CRO in country who has extensive experience helping companies operate there. We're also going to be submitting the same data package to the United States for an early feasibility study. This will be 2 to 3 subjects in the US. We've engaged with the FDA several times on mapping out what this looks like, as well as the data that's going to be required to support the submission. We're targeting 10 patients with heart failure with preserved ejection fraction plus PVR, very similar inclusion-exclusion to the first-in-human, and we anticipate submitting the same data package in both places. From a value creation timeline perspective, this slide shows kind of where we've come from, what we're doing now, and where we're going. The company came to MD-Start in 2020. Seed funding was invested, and with that, the company moved through all of the early stage work that we needed to do in terms of IP generation, assembling a team, building our plan for our clinical studies and our target patient population, developing the catheter, and freezing the design. We have yet to execute, or rather, we're midstream executing our preclinical safety study and are working on our design verification testing. We anticipate wrapping these up by the end of the year. We are raising a Series A financing. This will support our clinical program, first-in-human study, and early feasibility study, as well as any device iterations that are required following that clinical work. And from there, we would move forward into randomized control trials to prove the efficacy of the device. To wrap up, elevated pulmonary vascular resistance brings significant additional mortality and hospitalization risk in those patients with heart failure. There are no approved treatment options in this patient group. Early data from other devices repurposed from other indications indicates that this is a pathophysiology that can be affected by a procedure. We have foundational IP, which enables development of a built-for-purpose device. We've frozen the design of our catheter, and we're doing our preclinical safety studies. We're developing within MD-Start, the premier European medtech accelerator. We have an experienced team in place, and we're raising a new financing to support our clinical program. Thank you for your time and attention.